Okay, so thanks for staying around. This is going to be a little bit different from most of the talks, perhaps, although as Serena said, maybe they're all different. Red pointer, is that what we got? I may just point at things. Yeah, it's okay. Nah, I'm going to point. I'm going to use a solid state pointer. So, ooh, I like stick. Sticks, yeah. Okay, good. So my name's Ron Walsworth. I'm at Harvard and Smithsonian. I'm going to, now it's okay. I'm going to use this. Thank you very much. So don't get out of line. And I'm going to tell you about nanoscale magnetic imaging using NV color centers in diamond, nitrogen vacancy color centers in diamond. This talk is really going to focus on a little summary of the basic physics ideas that underline why these color centers are good for magnetic sensing, a little bit of the history, and then some particular subset of the applications. There are a wide range of rapidly emerging applications, particularly some bioimaging type applications, not exclusively, but mostly. I'll make some allusions to some other types of applications as I go along. But with limited time, that's what I'm going to focus on, and mostly stuff that my group or some of my collaborators have been involved with, just so you understand. Okay. So this is, I could boldly say, a new field in terms of making quantum sensors using these color centers in diamond. And it got started with some brainstorming that Misha Lukin and I did in late 2006, inspired by a question that Dima Budker asked when he was visiting Harvard and at that period. Misha, and there's other people in the world, Jörg Rocktrop, Fedor Yelechko, David Oshlom, Ronald Hansen, a series of other people, had been looking at these color centers, these nitrogen vacancy color centers, for quantum optics and quantum information applications, primarily in the late 90s, early 2000s period. And then, eventually, scientists think about it a little bit more, we broaden our minds a little bit, and we realize that they're actually very good sensors for things like magnetic fields. And later has come the ability to sense electric fields, temperature, et cetera. Okay? So it turns out that this activity is grown greatly. This first kind of proposal, if you want to call it a proposal paper with no experimental data, and then these two first experimental papers have 300, 400 citations, something like this, and there's huge activity, lots of groups working in this field, giant funding programs in multiple countries, people getting hired, et cetera. So hopefully it has some value. And the value is, at least for the magnetic imaging, is perhaps best summarized, although too simplistically, in this plot, which shows magnetic field sensitivity in the wonderful units Tesla per root hertz. Numbers getting smaller mean more sensitive, versus spatial resolution of your detector or your imaging system, where smaller numbers mean higher spatial resolution. These are not the only two variables one would care about. You could talk about bandwidth, field of view, many other things, cost, feasibility for different types of applications. And I'll mention some of those things as we go along. But these two things, and these are fuzzy blobs drawn in PowerPoint, so they're very generous regions, but this kind of tells you the basic story, which is that these color center, these MB color centers in diamond have a combination of magnetic field sensitivity and resolution, while in a very robust, practical, convenient, easy-to-use system that is better than squids and goes into regions which are heretofore unexplored or unavailable with any other technology, except I could have put magnetic resonance force microscopy working in little niches, and there's some other excellent but sort of more niche technologies. So it's a combination of the sensitivity and resolution together with this ability to have optical detection operating over a wide range of conditions, room temperature, low temperature, high temperature, pressure, acids, all sorts of different conditions. So very robust systems, easy to use, compatible with biological things. Also enable electric field and temperature sensing, and the community is working on adding different sensing modalities to beyond these few. If you look at what the magnetic field would be from a single electron, if your sensor was about 10 nanometers away from that single electron, it would appear approximately where that dot is. And NV diamond has been used to sense individual electrons at room temperature. And in fact, in Amir Jacobi's group at Harvard here, assisted by some of the other diamond groups at Harvard, so I'll say we've been able to image the 3D position of individual electrons and solids to angstrom scale resolution using diamond at room temperature. No other technology can do that. The field, if you were, say, 1 or 2 nanometers away from a single nuclear spin, like a proton, the field would be approximately that value. NV diamond has not yet been able to map and sense individual nuclear spins and map their positions, although my guess is it will probably become possible, no guarantee, but probably within a couple of years. the community, including my group, has been able to sense groups of nuclear spins that contain effectively like 100 nuclear spins in them, polarized nuclear spins. It's a sample that's a few nanometers across that might actually have 10,000 nuclei in it, but due to statistical polarization, the typical value of the magnetization is equivalent to that of which, as if you had, say, 100 nuclear spins. So that's what I'm talking about. So that's the kind of state of the art. I'm going to talk a little bit about that as we go along. All right, so that kind of gives you some examples of some of the benchmarks of what's impressive sensing. There are things that you would want to do with coarser resolution and higher sensitivity, but that gives you some of the examples. Okay, so I'm going to go through a few slides here to kind of give qualitatively, with a few numbers, some of the basic ideas that underlie sensing with diamond. We're talking about these individual and then also ensembles of nitrogen vacancy color centers in diamond. So here's a cartoon of the carbon lattice that makes up diamond. The NV center is literally one nitrogen atom that replaces a carbon and one missing carbon atom, sometimes called a vacancy, adjacent to each other. This is a common defect. There are many defects in diamond, hundreds of them, different types. Nitrogen is one of the most common impurities that you find in naturally occurring diamond, and you can grow diamonds in the laboratory and make it have nitrogen. Nitrogen is next to carbon on the periodic table. It's a little bit bigger, but it can squeeze and replace a carbon and have one extra electron without much trouble. Energetically, a vacancy next to a nitrogen is favored. Once they find each other, they like to stick together. So you typically have some process to grow in or implant some nitrogens, make some vacancies, often by irradiating the diamond with electrons, knocking out some carbons, heat the diamond up to 800, 900 C, anneals them. The vacancies get mobile. They find the ends, and they create these NVs, and they like to be together. Also energetically favored is to capture an additional electron, and so it's actually an NV minus. It's a charged, localized electron cloud. It is not a pink ellipse like I've drawn there. It's a much more complicated structure, but this is what I can do in PowerPoint easily. And if you saw the actual wave function, the theorist of you would be excited and the experimentalists would be bored, but it's a complicated wave function because affected by the lattice here. Anyhow, simplistically, it's a many-body interaction, so this is not correct, but I'm going to say it anyway because it gives a nice physical picture. You have an extra electron from the nitrogen, an extra electron that's added from the lattice, two electrons together. The dipole-dipole interaction, magnetic dipole-dipole interaction between them is going to lead to a level splitting, a ground state spin level splitting between the ms equals 0 and the ms equal +1 or -1 levels. And also, as I'm going to show the energy level structure soon, there's ground state spin triplet, because that means spatially antisymmetric for these two electron spins being farther away, and so the electrons, and therefore the coulombic interaction lowers their energy compared to the electronically excited spin singlet states, which have a more symmetric wave function. The measurement protocol, obviously these are cartoons, the measurement protocol typically involves illuminating with green light, manipulating the electronic spin in the NV center with microwave pulse sequences, which are analogous to what's done all the time in NMR, and then looking at the red light, the red fluorescence that comes out. Hit it with green light, manipulate it with microwave pulse sequences that you control, and then measure the intensity of the red light that comes out the fluorescence. That's it. Of course, you have to control a variety of things in your system, but it's hit it with green light, microwave pulses, measure the red light. And the intensity of the red light, if you do it correctly, is a measure of the local magnetic field, as I'm going to describe to you. So it's an atom-like defect in solid. It has an s equal 1 electronic spin. It can have, because of this large 5.5 electron volt indirect band gap in diamond, these deep donors are very uncoupled from the lattice, other than just general motion of the lattice, vibronic motion, which shifts the energy levels around. But the spins are largely uncoupled from the lattice. So you can have remarkably long coherence times, millisecond coherence times for electron spins in a room temperature solid. That's a robust, common solid. Because they are electron spins, they have a large Zeeman shift, Bohr-magneton Zeeman shift. So the fact that energy levels move rapidly with magnetic field lets you know why it's a good magnetometer. The fact that you can prepare the spin states, as I'll describe optically, and read them out optically, makes it a convenient high signal to noise system. Okay. Operates under a wide range of conditions. Here is a cartoon of the energy level structures, the energy levels that are relevant for the NV center, not the many-body physics of all the carbon atoms and things like this. Ground state triplet, ms equals 0 and ±1, split by 2.87 gigahertz due to the You can think of it as the dipole-dipole interaction between the two electron spins that are making up this NV center. Okay? Separated by optical transitions from electronic excited states and two metastable singlet states here. So the levels are going to shift rapidly with magnetic field. There is absorption of green light and radiation and fluorescence in red light. There are also non-radiative, non-spin preserving transitions here that are key. And I'll describe that now. So if you want to do some coherent spin manipulation with the MV electronic spin in these color centers, there's three basic steps. I've already said them, but I'll go through them again. One is illuminate with green light and prepare your states. You get a kind of spin polarization, a spin state preparation into the MS=0 state within about a microsecond. Why is that? Because when you're in the if the MV happens to be in the MS=0 state, it can absorb green light. In about a peak a second, it comes to the bottom of this excited state vibronic continuum and then will fluoresce in the red in about 10 nanoseconds with a very high probability. So it's close to a cycling transition. Absorb, fluoresce, absorb, fluoresce, absorb, fluoresce, over and over again. If it happens, however, to be in the MS=+1 or -1 states, it can also absorb the green and fluoresce with about a 60% probability and has about a 40% probability on a longer time scale to undergo non-radiative, non-spin preserving decay down to the MS=0 level. There are some various weak cross relaxation terms, but this is the dominant processes. So that means if you're in MS=0 to begin with, you stay in MS=0 and you just give out high rate of fluorescence. If you're in MS=+or-1, you'll fluoresce a little bit, but then you'll decay into MS=0. So that means step one is wait about a microsecond, put on the green light, and you'll end up in MS=0 no matter what spin state you started out with. That's your preparation step. Then comes step two. You do whatever coherence you want to do with resonant or near resonant ESR pulses. If you want to do a magnetometry sequence, you do one thing. If you want to measure electric fields, you do a different thing. If you're interested in some quantum information protocol, you do a third thing. There's a wonderful array of pulse sequences, and they're being improved all the time. Paola Capellaro will give a talk later this afternoon down at the physics department, is it? Yeah. And so I recommend you go there, and she's wonderfully intelligent and inventive and is doing some wonderful things in quantum control, often using NV diamond. We collaborate closely. Then finally, when you've done your protocol, whatever it is, in our case magnetometry, you can, let's say you tip over, and I'm going to actually move along so you can actually look at the, let me just move forward here for a second. Well, okay, I won't move forward. Let me just say, let's say you prepare in MS=0, you give a pi/2 pulse with your microwaves. You let the spins process in some magnetic field you want to sense, and then later with an additional pi/2 pulse, you project along the axis, either this way, or some mixed state, and then you measure the level of the rate of red fluorescence that comes out. Perhaps you have to repeat that a few times if it's a single NV. Perhaps one measurement's enough if it's an ensemble of NVs, and the intensity of the red light gives you the net phase shift. It's proportional net phase shift. So this is a plot of averaging several scans with a single NV. Red is if you started out in MS=0. Blue is if you started out in MS=1. Over the time scale of a few hundred nanoseconds, the fluorescence grows in the MS=0 and basically levels off. MS=1 has a lower rate of fluorescence for a while, and then eventually on long time scales, as it decays back to MS=0, it reaches the same fluorescence rate as the MS=0, because you always end up in MS=0 if you wait long enough. But in this intermediate time scale in here, the level of fluorescence is an indicator of which spin state you were in. So these are names of a few of the many people who contributed a great deal over several decades to figuring out this basic physical picture here. There are many people. I'm just hiding some people that matter the most. My hero, if you will, is Neil Manson from Australia. He did more work than anyone else, in my opinion, to figure out this basic energy level structure here and do some of the underlying physics which leads to this, and also prove it experimentally. I'm giving you, not surprisingly, a simple picture of what's going on. There are many other important issues in practice. The NV center is here, but there can be other spin impurities. In addition to the weak couplings of the lattice and the magnetic fields you might want to sense, there can be other spin impurities like carbon-13 nuclei, which have a nuclear spin, 1/2. Paramagnetic impurities like nitrogens or other impurities, electronic spins in there, and they can be processing and interacting and flipping and creating magnetic field noise that can be sensed by the NVs. So in fact, there's a whole community of people interested in studying those spin baths, flip-flop interactions, spin diffusion, interesting things going on in spin best. We've done some of that work. Many other people have too. There's also people who say, well, let's get rid of that spin noise. We want to have low impurities or dynamic decoupling sequences to basically protect our NV and isolate it from the spin bath fluctuations, et cetera. So that's a whole bunch of things that are going on as well. You can also think about using these spins in the environment for sensing, environmentally enhanced metrology. And there have been stepping stones taken in that direction, but as yet, all the sensing work I'm going to describe and what I've described already is semi-classical. But I think we will get to start using non-trivial, let's call them quantum information inspired or derived protocols in some of the sensing protocols, in some of the sensing methodologies fairly soon. For example, you could make a measurement with an NV by an appropriate C0 analog interaction and hyperfine interaction, map some of the phase shift that you've measured onto the nearby carbon-13 nucleus, which has a much longer both spin coherence and T1 time. Then turn off that interaction with a smart, or effectively turn off that interaction with a smart pulse sequence, and then make another measurement with NV, and then repeatedly map the phase shifts that you've measured onto the carbon-13, and they accumulate, and you basically can use it as a register to store that information. Misha Lukin's group and some others have done some stepping stones in that direction, very nice demos, not yet using them for actual metrology and sensing, but I think that will, to enhance it, although the theoretical protocols are clear, that will probably happen. You could also use the electron spins in your environment, which have nice Bohr magneton magnetic moments like your NV, as sensors. They don't have all these super cool optical properties where if you hit them with green light, they spin polarized, and then you can eventually read out the spin state with red fluorescence. They don't have that, but they have large electron, they have large Bohr magneton magnetic moments, and they can couple strongly to the NVs as well. So how about transfer the spin polarization from the NV to the ends, address, or the electron spins that are in the environment, address them with pulse sequences, let them do some sensing, map the information back into the NVs, and read it out. So that is on the plan, the master plan. The community is working in that direction. My group and others have done some of the stepping stones of transferring spin polarization from the NVs to the ends and that sort of thing. So that will probably happen over the next few years. There are 4 crystal axes within diamonds, so the NVs can be found along 4 crystal directions. That's both a good thing, and that it gives you vectorial magnetometry. With appropriate application of static external uniform fields, you can choose, and then knowing what frequency your microwave pulses are at, you can choose to operate on one NV axis as opposed to another. However, no matter what you do, all the NVs are fluorescing. So even if you're looking resonantly at one axis, the other 3, sometimes you can do it so that you're looking at 2 rather than 4, if you set the fields up right. The others are providing photons that are background photons that don't have information in them. So it's a kind of reduces the contrast of your measurements. So those are grungy experimental issues that we have to deal with. Therefore, sometimes the community is interested in growing NVs or having NVs created in the diamond that aren't equally populating all 4 directions. And there's some work in that direction. We've done some work in my group together with Element 6, realizing NVs along 2 of 4 axes, and some other groups are now getting along 1 axis, et cetera. You lose some vectorial capability, so there's pros and cons. And there are other issues. You always want your geometry of your diamonds to be the way that you want them for your particular application. This repeats what I said before. It just says that there are different types of pulse sequences. For example, Ramsey sequences for measuring static DC magnetic fields or slowly changing magnetic fields. Spin echo and spin echo extended like dynamic decoupling sequences for AC fields. Other types of sequences as well. I'm not going to dwell on them too much, just to let you know that there is a plethora of them. There are many applications. Everything from buried ordnance probe to nano MRI and live cell thermometry, all sorts of stuff. And this is an incomplete set. It's just sort of what came to mind as I spewed them around us on the PowerPoint. But there's lots of things going on and rapidly evolving, some very basic, some very applied. Here's the community of people at Harvard who are working on diamond in one way or another. Misha Lukin I've mentioned, Amir Yacobi I've mentioned, Hong Kong Park we collaborate with closely, who's in chemistry and physics. Marco Longcar is an expert in nanofabrication and nanophotonics. He's done some amazing things in making diamond nanopillars, et cetera, with NVs in them in the applied physics and engineering. Jeff Lichtman is a leading neuroscientist with whom I collaborate. Evelyn Hu is an expert in material science and also does some nice work in diamond. There's a large group of people that we collaborate in various combinations outside of Harvard. Dima Budker, I mentioned. Stefan Hello is a leading super-resolution optical imaging innovator and bioimaging scientist. Dan Twitchen, who's at Element 6, expert in diamond growing. Jörg Grobschop and Fedor Jelechko, who've been pioneers in MV diamond physics and in other applications. Phil Hemmer, similarly. Paolo Capellaro, I mentioned. Very creative and intelligent junior faculty member at MIT. Hako Lee, I'll talk about some of the work we've done together with him. He's at Mass General Hospital and is developing tools for doing bioassays and studying circulating tumor cells. We've done work there that I'll talk about. And Arash Kameli is a cell biologist with whom we've done work in magnetotactic bacteria, doing basic cell biology using NV diamonds, and he's at Berkeley. There are many modalities of these sensors. You can have nanodiamonds. You could probably guess the red dots are cartoons, okay? But this gives you the basic idea. There can be nanodiamonds with no NVs in them, one NV in them, many NVs in them. There are other kinds of color centers other than NVs, but don't have time to talk about them. You can have atomic force microscopes. This is built in Amir Yacobi's group, developed together with Marco Loncar's group to have this diamond nanopillar with a single NV about 10 nanometers from the surface. So with the piezo's, they can control and scan over the surface and map the magnetic fields from individual electron spins and other sources. Amir and I are collaborating closely now on studying 2D magnetic materials. I think there's going to be huge applications in terms of this technology towards basic condensed matter physics and material science. Don't have time to talk about that today. You can have NV centers which are very shallow, like a few nanometers below the surface of diamond, and then sense small numbers of some sample, like nuclear spins, on the surface. And I'll talk a little bit about that kind of work. That's been very successful. One can also have ensembles. You don't have to have just one NV. You can have many NVs to boost your signal to noise, pack them in at high density into some geometry of interest. For example, in this cartoon is meant to illustrate a macroscopic diamond chip, not that big, but let's say a few millimeters on a side, up to a centimeter on a side, where we've grown or implanted a high 2D density of the NVs in a shallow layer, maybe 10, 20, 100 nanometers thick or deep, depending upon what our application is. Then you can place magnetic samples of interest on the surface of the diamond. All those NVs can sense the pattern of magnetic fields coming from whatever the source is and be imaged onto a camera, like a CCD or CMOS camera. You can then have a pattern of magnetic fields. It's a 2D magnetic field imager. We call it the imager, basically. You can also measure the change in time of this sort of thing in a video rate. Here's some examples, a biased set, my opinion, of some key application results to date, particularly recently. Biased, you'll see the word Harvard appears over and over again, okay? But it is my opinion. So, MRI of single electron spin in a solid with less than 1 nanometer resolution. That will be published in Nature Nanotech shortly. There was being able to see an individual electron spin outside of the diamond, and it was in Nature Physics last year. This is work led by Amir Yacobi's group. Doing NMR of something like 100 nuclear spins, proton spins, in few nanometer organic samples. Published, this is Jörg Grobschop's group at Stuttgart, Dan Rugard, IBM, in Science last year. We have new data extending this, and I'll show you briefly in a few minutes, to other nuclear species, fluorine, phosphorus, etc. Doing magnetic field imaging of living biological cells with subcellular resolution. I'll talk about that in a few minutes. Published in Nature, done by my group last year. This is led by Misha Lukin and Hong Kong Park's group, taking nanodiamonds, putting them in living biological cells, and using the ways that the energy levels shift with temperature to do optical measurements and probing of intracellular temperature changes, which they then would change purposely by having additional nanoparticles, gold nanoparticles, put into the cell, and then with a laser, you can have the heat locally inside the cell. They're able to map sensitively the changes of temperature and use this as a measurement feedback tool to feedback towards controlled killing of cells or changing of affecting the DNA or catalytic processes. A very promising technology. Collaboration largely Misha Lukin and Hong Kong Park's group. I played a little bit on this role on this too. Single biomolecule sensing, having biomolecules which are labeled with electron spins attached selectively to the surfaces of diamond with single NVs and being able to image the presence of individual molecules one at a time. We've also done MRI of tumor cells as a bioassay, a high throughput bioassay of large numbers of tumor cells, and we're planning to expand this to pathogens and other kinds of rare cells. This is a needle in a haystack problem. You have a few things you want to find or identify whether they're there amongst a large number of normal cells. Can you find them? That's the question. And we think diamond can do far better than any existing technology, and that's from my group, and I'll talk about that as well. Okay, so now some more cartoons and then some data. So if you want to do this sort of nanoscale NMR spectroscopy and imaging with NVs, there's at least two modalities. One is to have one single NV that you're probing, and then some sample that's a layer on the surface. There may be some thick layer of stuff on the surface, but one single shallow NV will be sensitive to the magnetic fields that come from the spins that are only within a distance that's a couple times the depth of the NV below the layer. There are fields coming from all these other spins, but they're so weak that the vast majority of the field comes from this little, sort of little dome in here. Right? It can be nanoscale. So the deeper the NV, the larger the region you're sensitive to. The shallower the NV, the smaller. Right? Dipolar fields fall off as 1 over r cubed. You have to integrate over all the spins that are there. So if you look at conventional NMR and MRI, you use inductive detection. The sample volume is at best a few microns in size, more typically more like millimeters, centimeters in size. In your body, when you go to an MRI machine in a hospital, you get a little bit better than 1 millimeter spatial resolution. Here we're talking about nanometer resolution. And you operate at high magnetic fields. With this NV technology, with optical detection, you can be at low magnetic fields, anywhere from essentially 0 to high field if you really want to. You're dealing with statistical, not thermal polarization. You can have these very small sample volumes. A second geometry is this imager geometry with an array of NVs, all shallow. And now each NV is sensitive to some little region, but you can basically do 2D imaging and sensing of magnetic fields a few nanometers off the surface and whatever patterns are there. So here's some examples of data from a single NV case. This is looking at a particular mixture, well, a particular type of material that contains both protons and fluorines, primarily fluorines, often called PFOS, placed upon the surface, dried, and you can measure the spectra as a function of magnetic field. Here's the chemical structure of both the fluorine and the proton. Fluorine, proton, et cetera, NMR signatures. You do this by sensing with the right kind of protocol and sensing the Larmor precession of the nuclear spins. There's both protons and fluorines in the sample. And if you do things appropriately, the Larmor precession of the statistical polarization essentially causes an appropriate phase shift in the NVs, which you then can later read out optically. If you do it as a function of magnetic field, and find where the centers of these different spectral lines are, you find something that agrees with the book values, so you know you're sensing the fluorines and the protons. Okay, it's not very interesting spectroscopy yet, but it's the beginnings of doing spectroscopy, just actually seeing the signals, seeing that they are from fluorines and protons, and seeing that you can do them in very small nanoscale samples. You can go a little bit, you can do alternative things like have some structured pattern on the surface. We've also done this with ATP that has phosphorus in it, but for sake of time, I'm not showing you all the data. Here's something where we create an SiO2 mask, a structure that's about 100 nanometers off the surface, and then there's bare diamond here. We put some of this PFOS fluid, let it dry on the surface. The stuff that's on top of the mask is so far off the surface from the NVs that the NVs don't see the magnetic field. The stuff that's in contact with the diamond here, they sense. So if you look in the blue region, you see both the protons and the fluorines. If you look in the red region, you only see the protons. There's protons, unless you purposely clean surfaces with acids, you end up getting organic layers of protons everywhere. Whereas fluorines you would only get where you put the material. So you then can make an image, essentially a map of how much fluorines you have as a function of position, now with about half a micron spatial resolution. Again, this is a very boring sample, but it has spatial resolution that's far beyond the ability of any other magnetic imaging technology at doing things under ambient conditions, any conventional NMR or MRI. So it's the baby step towards doing nanoscale MRI. Okay, so while we're at that nanoscale and doing these baby steps with rather boring samples, we've also done some things with not very boring samples, things that I personally find now that I've started to learn about them, these biological samples, quite interesting. These next two experiments I'm going to tell you about both use this wide field imager. So the shallow, high-density 2D layer on a large diamond chip where you plop the samples down on the surface. Here's an actual photo of the experimental apparatus, control electronics outside, not shown, computer, etc., operator not shown, but it's relatively small and simple and could easily be engineered into a modest box and commercialized. In fact, that's one of the things that we're thinking about doing. It involves a diamond chip with the biological sample. The first case I'm going to talk about is bacteria sitting on the surface of the chip. The green light comes in from the side. We purposely do it below the total internal reflection angle so that the green light does not propagate through the sample, frying the biological sample so they can stay alive and viable. And then we collect the dim red light out into onto a, in this case, CMOS array. We can do wide field imaging upwards, as I'll show you eventually, of a millimeter. Currently, we should be able to get to centimeter field of view with about 400 nanometer spatial resolution and measure for the first time the magnetic fields that come from within biological cells. The first system that we studied were these magnetotactic bacteria, which are pretty damn amazing things that I didn't know a lot about until about 2 years ago. But get this. These babies evolved, the biologists tell me, about 2 billion years ago. They're very common in prokaryotes. If you remember your high school biology, Mark's probably studying biology all the time, but if you remember your high school biology, which I barely do, prokaryotes are not eukaryotes, right? They have no cell nucleus. They're extremely simple bacteria, single cellular, no nucleus, no sensing apparatus, etc. But they have evolved a way to sense. They naturally synthesize magnetic nanoparticles, which are more regular in size than humans can synthesize. They are naturally solve the problem of making the size of magnetic nanoparticles just above the blocking volume, where they do not act at room temperature in a super paramagnetic way. Make them a little smaller, the magnetization vectors start hopping around due to thermal fluctuations. Pretty amazing, huh? Then they align them along a protein filament. They do this all naturally. Makes me want to think about intelligent design, but I shouldn't say that. Hopefully no one's recording. Oh, wait a second. And along a protein filament, and with the magnetization vectors in each nanoparticle aligned in such a way, opposite in the two hemispheres, northern and southern hemisphere, opposite in the two hemispheres, so that they want to follow the magnetic field vector down, because they're anaerobes, and they like to be low in the water where the oxygen levels are low. There are many species. They're very common. They exist in Fresh Pond, probably in Walden Pond, where Suzanne loves to swim, and other places like that. So they form the chains. Some of them do this magnetite. There's other types of magnetic nanoparticles. They're not all exactly the same. Not all species do it exactly the same. A lot is known about them, but a lot is not known about them. And in fact, there's a healthy biological community that would like better tools to study the magnetic fields in living bacteria, sometimes in dead ones too, that come from that are coming from within the cells. And we have provided that now with our quantum-derived diamond technology. There are many questions that they want to assess. For example, the genes, some of the genes which are believed to lead to this and control this are also found in you and in higher organisms. Not just you. Special, very highly evolved organisms. Even me. Even less evolved organisms like me. Okay? And intermediate ones like, well, okay, well, I won't point. That's rude. That's rude. No, I didn't point. Okay. Why? They're not, I don't think we're synthesizing magnetic nanoparticles. That's not what's believed. It's believed that some of these genes have been co-opted for regulating iron and processing and other metals within the body. So they're a nice model organism for studying these things. People also would like to be able to put the genes into even more robust, highly productive other bacteria to basically make little factories to manufacture large numbers of magnetic nanoparticles. These names look more German, although there's a German PI, there's a Chinese guy here. Here's a paper that came out a couple weeks ago in which they're beginning to do these things. They took the magnetosome island, of the genetic island, the genes out of a type of MTB, magnetotactic bacteria, put them into a very robust, different type of bacterium that does not grow these things, and it began growing them. Obviously, these are cartoons, but I want my MTB, yes, exactly. For a number of years, people like Cam Moeller, very clever professor at Stanford, have been using squids to study the magnetic fields that come from MTBs and adding value to that biological community. However, the resolution of the squids are only about 5 microns. The MTBs are about 2 microns long. You can't see the intracellular fields. Also, to get that kind of resolution, everything has to be cryogenic. You could have a bigger squid stood off and have the MTBs alive, but then you're not going to see individual bacteria at all. And it only senses the one component of the field, not the vector. So this doesn't, other than saying, yep, they've got a magnetic field, we can sense it one bacterium at a time. Once they're dead, they can't really do a lot with that technology. With our imaging microscope, we can do wide field of view images. Here's a bright field image where each squiggle is a bacterium, and we can measure the magnetic field patterns that come from individual bacteria. If you zoom in, you can see the fields that come from individual bacteria. Here's side by side comparing the bright field image and the magnetic field patterns that you get from individual bacteria. It works very well and relatively easy. And they're alive. Sometimes we do them alive, sometimes we let them dry. Depends on, in many cases, letting them dry on the surface is fine. After you've let them be alive and do whatever they want to do, that lets more stable. In other cases, they're alive, and we image them when they're alive. In this case, for example, it's dry. Because here's an example in which dried out. It was alive, and then we let it dry. Why? We wanted to say, okay, great, you can see these fields. They look kind of nice. That stuff looks good, and it's a dipole-like pattern that's consistent with what people expect, kinda, but let's be a little more quantitative. Let's take one bacterium. We did this for more than one, but let's take one. Measure with the diamond magnetic field imager the BX, BY, BZ fields. Then take that same, now dried, you'll see why we had it dried, so that we can take it to an electron microscope. Determine where the magnetic nanoparticles are, then given the known magnetization, the typical magnetization of those nanoparticles, calculate what the magnetic field should be, given the location of those nanoparticles, and that's the fit. There is a couple free parameters like exactly how far off the surface they are, but there aren't too many free parameters, and this is what we calculate from nanoparticle position and what the field should be, and this is what we see from our diamond imager. So we did this a few times. Consistent results. It isn't too surprising, really. You have this magnetic field source. It should make a field. You have an imager which can image those fields, and you get the right values. How about Oh, electron microscopes, of course, have much better spatial resolution. Okay, so you can get to much higher spatial resolution with electron microscope. Then when we calculated the fields, we purposely coarse-grained what we mapped here to be consistent with what we would get from our imager. You bend like 400 nanometers, 400 nanometers. All right. So we don't have to plot it in Gauss, but we did plot it in Gauss in this case. So yes, because once you have these nanoparticles up so close to the surface of the NVs, the nanoparticles are about 50 nanometers. They're close to sitting on the surface of the diamond. The NVs in this case are about 10 nanometers deep. So you have in one of those nanoparticles, I want to say 10 to the 4 electron spins, but I'd have to go check. There's some large number of ions in them, but it's something like that. And so it's a very large local field. Of course, the field on length scales large compared to the size of the nanoparticle. It falls off approximately as 1 over r cubed, which again, if I went back and talked about other kinds of magnetometers, one of the great advantages of diamond is just you can get close to the fields. All these wonderful atomic magnetometers and other things which have femtotesla or whatever sensitivity, that's great, but there's lots of noise that can swamp that level of sensitivity. So the only way you use a femtotesla magnetometer is by putting it in magnetic shields. And then you have to have something that you can bring in close to it. With a diamond magnetometer, what you do is you make your fields large by coming up close in direct contact with it. Then you can have all this background noise, who cares, right? Because your fields are big. But they're only big when you're close, if they're dipole-like, okay?
So when the bacteria are migrating, are they sensing the pull and orienting themselves?
Yeah.
Or is the magnet actually...
They have little flagella, 1 or 2, they're swimming, and it slightly biases the random walk. Now one of the things I haven't learned well enough yet, but I'm planning to, is to really kind of probe that biophysics of it. It's a kind of information processing. It doesn't have, it's not through neurons, it's just a physical thing. You've created one physical force, a mu dot or an m dot b force, right, from the, and some diffusion that's moving it around, plus a flagella that's kind of pushing it in one direction. And over time, more often than not, they head down a little more on average than up. And given their limited ability to do anything, unless they, to do anything else unless they became eukaryotes or something like this, but given the limited tools that they have, it's really remarkable. So you could argue it's a kind of information processing, a kind of physical information processing, as physicists would say. You know, any physical system that's following the principle of least action, whatever, is processing information. So are these little bacteria. Okay, sermon over on that. So let me say, here's an example of other things we did. Okay, and I'll try to say this quickly. We took, the optical means diamond magnetic field images. These are vectorial patterns of the magnetic fields as determined from the kind of images we had before. From that, we went back and looked at where the divergences were large and modeled where the magnetosome chain should be. And that's shown in yellow. So that's concluded from our model. We make measurements, and then we use a model to infer where the magnetic nanoparticle chain should be. And then we go and we look at the actual electron microscope images and see where the magnetosomes actually are, magnetic nanoparticle chains. And we find very good agreement. Okay. So I'll skip ahead because I think Irina wants me to stop. And so I will say that we've also applied this to tumor cells with magnetic nanoparticles labeling the tumor cells. It's a rare cell problem. Let me just skip ahead to this kind of image. So here's an example now, millimeter field of view. The pale circles are normal cells that are not labeled with nanoparticles. We've spiked a few tumor cells, breast cancer, mortal line tumor cells, which have been labeled with magnetic nanoparticles and purposely labeled with a fluorescent dye, each going to the same biomarker, and spread them out on the surface of the diamond. And we see that we can see where there's no cells, we can see normal cells, we can see bright things which indicate tumor cell, and then we make a magnetic field image. So that's just a fluorescent image. And then we do a NV diamond magnetic field map. And everywhere we see a bright cell that indicates that that it's a tumor cell. We also see a magnetic field pattern. Sometimes there's a couple minutes between the images and there's a slight position shift or something. But basically, for every circle that we see here, we see a magnetic field image there too. So that tells us that with the magnetic field imager, we can label these these cells with nanoparticles and see them. Just like you could see them with fluorescence. You say, okay, that's great. You can see them here, or you can see them here. That's nice, but is there really anything that makes it better? This just shows that we can see multiple different types of cells and multiple different types of biomarkers. I won't go through the data given limited time. But here is something that shows we can do things that the conventional optical techniques cannot do. Because magnetic fields will penetrate through thick biosamples like blood, whereas fluorescence from some fluorescent dye will be absorbed or scattered. And so if you try to see bright field transmission where there's one cancer cell looking at the fluorescent dye in a blood sample, you don't get any signal, whereas the magnetic fields, not surprisingly, pass right through the blood, and you can just collect the fluorescence from the MV diamonds coming to the imager. Okay? So this indicates an important utility, wide field of view, potential for high throughput. This is less than 1 minute per image. We're upgrading. We think we should be able to get down to less than 1 second per image, where this basically be flowing through image. You have large numbers of cells. So really the utility of this sort of technique will be not only that it can do things that other techniques can't do, but that it can scale up and you can go high throughput. All this technology for labeling cells with magnetic nanoparticles developed by others, our collaborators at MGH. They were already trying to do this wide field magnetic imaging, but not using diamond, using arrays of Hall probes, which works fine up to a point. But as you start to scale up, the number of wires goes crazy, the heating, and so diamond, it's very easy to just scale up and make you need a little more optical power, but make your field of view wider and wider. This talks about how we're in the process of scaling up. I won't go through the details. I'll conclude by quick comments on speculative directions, if I may. One is to get to true atomic, for this NV magnetic imaging. One is true atomic scale MRI. Map every nuclear spin and chemical bond in single biomolecules, proteins or whatever. Is it going to be possible with a very shallow NV center, very strong magnetic field gradients, etc.? It's a nice motivating direction. It's going to be very challenging. One has to do things like immobilize this and knowingly immobilize it while you do your sensing. Maybe you can have nanodiamonds or nanopillars going in there. I think it's worthwhile. Some of the DARPA programs and other things that are funding some of us are supposed to head in this direction. We're doing a lot of useful things already, but this is going to be very challenging. Another is a kind of nanoscale functional MRI, functional in the sense of neuroscience. Being able to have grown cultured neuronal networks on the surface of diamond where axons are firing and sensing not the electric fields, but the magnetic fields that come from neurons. We've modeled the sort of fields. We think we should be able to see such fields, and we're in the process of building up to do this kind of 2D neuroscience. So getting imaging culture, that means grown on the surface of the diamond neurons in a couple of years, is probably possible. But you're not really, and that's nice, but you're probably not doing anything that really turns the neuroscientists on, who we talk to, until you're at this level, imaging whole brains of simple animals, like C. elegans worms or larval zebrafish. These things are quasi-two-dimensional and transparent, so it is possible to imagine them sitting on top of, maybe they're swimming and functioning, your NV diamond imagers here and measuring all the firing neurons, seeing the magnetic fields. Because they're quasi-two-dimensional, it's possible to imagine doing that. Will it be possible? Challenging, but maybe. How about an application or interface Borg style with a whole mammalian brains in some time that government officials think is infinity, you know?
Be assimilated.
I don't know. I don't know. I think yes, maybe, I don't know. That's my answer. Yes. How do I measure what?
How do you imagine that doing if you need to be really on the real, transparent, three-dimensional mammals?
Right. So one of the key things is how close these NVs can get to these neurons to be able. So People have sensed the magnetic fields from individual axons in cases that are, to date, who has sensed a magnetic field from a single firing axon? A couple groups only ever, and in cases which are not all that relevant to any of these things. Like they took, don't get confused here, but they took things like the giant squid axon, not the quantum squid, but the actual squid. They took a giant squid axon, put it through, now it's going to get complicated, don't let me blow your mind here, an actual squid, sensor squid in the squid, and then sense the magnetic field. It's pretty damn cool when you read this stuff, so don't laugh, it's pretty cool. But the size of these giant squid axons are so big, and the fields are so big that if you try to extrapolate to mammalian neurons and things like this, and I talk to biologists, they go, hell, we don't know. So we've got to do some of that work to then know how big are these fields actually in neurons of interest, to then do the scaling argument. If the C. elegans is, if you're going to be standing off a micron or so, or two, from the firing neuron from the NV layer, is that, forget it, the fields are going to be way too small? We don't know yet. That's why I'd say maybe. If I knew the fields were going to be large enough and with optimistic projections of what our sensitivity will become, that we could do it, then I would go from maybe to probably. But that's why I say yes, probably here, maybe, heck if I know here.
But the theory of the two-dimensional transparent mammals is all worked out already.
Clarity. It's kind of interesting, though. I get to learn things about squids and whatever. This, I think, would definitely work and have impact. This is to take human brain tissue, you can also have animal models, autopsy and biopsy tissue, where there are known to be diseased tissues with people with diseases like that, where there are known to be magnetic plaques that give off signals, and people already see them with squids, the sensor, not the animal, okay? But they see them at length scales, given the coarse resolution of squids, and under conditions which are not what they want. They think that these collections, unnatural, or no, that's the wrong word, non-functional disease state collections of extra iron are beginning at the kind of 10 nanometer to 100 nanometer scale. That's what they want to see. They need higher spatial resolution. Diamond can give that to them. And so I think this would likely work and have big impact. And then there's a general category of functionalizing nanodiamonds to go into cells and do useful things, either sense things, guide therapy. The result from the Lucan Park groups on the thermometry is already a step in that direction of being able to sense temperature. They could see a half a degree Kelvin above normal with a little bit of heating in the nanodiamond inside a particular type of cell, and everything is alive. They have an indicator of whether it's alive or not through a fluorescent dye. And then if they heat it up to about 4 degrees above ambient, as sensed by the MV, it dies. And so going beyond that to other kinds of therapy guiding and sensing in vivo, some of the things will work and some will turn out to be too challenging, but there's a lot of opportunity there. A lot of challenges, but we want to identify high impact applications. I think there's a long way to go in terms of improved sensitivity and measurement modalities. We're already having impact on certain fields like these bacterial. We've got some grants funded together with biologists using this sort of technology. The communities over there, like in the MTB area and others, are very excited about it. The people who are doing the things with Alzheimer's and Parkinson's are very excited about that sort of applications. We've even started a company to commercialize these things with our nice smiley faces, and some people working with us, including one of John Doyle's former graduate students who's an excellent scientist, Colin Connolly. We've won a phase 2 SBIR funding some of this work, which from DARPA, particularly the circulating tumor cell work, which tells me, along with the fact that other companies are going into this area. AtoCube has taken things like this atomic force microscopes with the diamond on the end, is commercializing it, and several other companies are doing too, that this is moving from our community out into the field rapidly, and that's a good thing. So let me end, I'm over time as usual, by thanking the members of my group, most of whom are still here. Anat and Nir recently last summer moved back to Israel. Nir has a faculty position at Hebrew University. The people who've worked very hard on these diamond projects. These are the people who are working on diamond things in my group. Chinmay Belthangadi and David Lesage are fantastic postdocs who are currently on the job market. I would extremely highly recommend these guys. They've gotten close to getting jobs at several places. The job market currently is tough. They've been on the short list at several universities. Outstanding people, very creative. David Lesage had the idea for doing the MTB experiment. I was wanting to push first to the neurons, but he was correct in saying, no, we should do something that's technically easier, but will also have impact on biology. Do that first. He started the collaboration with Aresh Kameli at Berkeley, which has then flourished, and was the lead author on that paper. So he's done a fantastic job. Chinmay has done some great work in terms of the more quantum side of things, doing some of these Hartmann-Hahn couplings to transfer polarization. There's some great people in my group, and obviously I'm selling them to the people in the room here, but they are really good. And I thank you for your attention, and you can ponder these beautiful pictures. These bag of jewel images are meant to indicate that there's a lot of opportunity, because if you take these are micron scale diamonds, and you stick them in a UV lamp, they give off beautiful colors. When they're red, that means they have predominantly NVs in them. Because, in fact, in a naturally occurring diamond that you've seen, or one that you've bought for your wife or girlfriend or husband or whatever, pet or whatever, you find that the pink diamond is overloaded with NVs. But many of the diamonds here are different colors. There are lots of other color centers, many of which are known in a kind of gross materials characterization sense. And I'm not criticizing them, but what I mean is they haven't studied them yet as individual quantum objects. So I think there's a lot of opportunity, and not just in diamond, but in other crystalline systems too, to basically take the wonderful levels of quantum control and understanding that we have in atoms and ions and other things, bring them into the solid state systems, the right kind of system, so we can get them out into the real world more effectively and interface with the real world. Okay. Thank you.
All right, so I guess...
If anyone wants a lab tour, we have 5 diamond setups down in the lab, downstairs, 2 more down in my labs that I have in the Northwest building down the street. We have imagers, we have STED super resolution systems, we have confocal microscopes. We got it. So you're welcome to come take a look. We get them from several places. More than any other, we get them from Element 6. Element 6 is a private company. They were the diamond research and manufacturing that's growing in the lab arm of the De Beers diamond cartel, which has controlled much of the diamond, not all, but much of the diamond market in the world for a century or so. Founded by Cecil Rhodes, a very interesting character, a little over about 100 years ago. So De Beers mines diamonds primarily out of Africa, as well as has Element 6. But Element 6 was recently sold to, and perhaps the whole De Beers cartel was taken over by Anglo-American Mining Company. But Element 6 has fantastic scientists who are at the frontier of learning how to grow diamonds with color centers and other defects in them in the right positions and having the right properties, long coherence times, et cetera.
Do you ever use natural diamonds?
We've used a few natural diamonds. That stuff there is grit that comes out of, well, okay, it's HPHT diamond that's then cracked up. But yes, some natural diamonds. And you can find NVs in natural diamonds too. But typically you want certain conditions. So rather than, well, I've got a pile of natural diamonds, eventually I'll find the one I want, that sort of thing. You have somebody make the diamonds for you.
That's the only one difference that you know that you have more accuracy.
Yeah, or others, the shapes are what you want, that sort of thing. Of course, they could take a natural diamond and cut it down too. Some of this is related to a dirt.
NVs right at the top too.
Yeah.
You don't want them in the bulk where they're going to fluoresce and give you background.
Some of this is related to a dirty little secret that I will tell you, which is that it's not so secret. The value of diamonds is much less than you think. What you pay for gems and things is an artificially inflated price. Because they control the supply and they've created a myth that they're very valuable. They're semi-valuable, but they make this myth. And then for your sweetheart or your sweetheart, now this is legal, your sweetheart, whatever, you can go and spend all this money. And if I didn't really love you, Jim, then I wouldn't spend those thousands of dollars. So they create that kind of guilt-based stuff. But they're generally not that expensive.
What limits the resolution if you talk about diamond ensemble? I understand if you have one little thing, you know exactly what it is and you can control this precision. But if you have an ensemble, how well, I don't see how, probably I don't see how you can address the resolution beyond fraction of the optical wavelength, because it's still all optical, right?
Right, so how we can get higher spatial resolution than that. I'll give you a couple examples. Let's say we had a diamond nanopillar that was 50 nanometers wide and had 8 NVs of the same orientation, 10 nanometers from the surface across that things like that have happened. And the optical fluorescence light is actually a lot of it channeled up the nanodiamond and out. That's one of the things that happens. Okay, then you'd say our spatial resolution in that sort of situation might be something like this 50 nanometer wide. And with piezo's, you know where it is with some accuracy. So that's something below the diffraction limit. They don't usually do the measurements that way. They usually use a single NV, but that's one way. Another way you could do it is with super resolution optical imaging, like STED and others, in which you can have multiple NVs within your dark spot if you want. Typically, you're going to do it 1 NV by 1 NV. I don't know if you know how STED works.
Yeah, I know how STED
Okay.
works, but my impression was that you cannot do much better than optical wavelengths.
Oh, no. So we've gotten down to 10, 15 nanometers. Yeah.
Oh, really? Okay.
Yeah, that's with single. We can have multiple NVs, but we look at one NV at a time. Now, another thing that we're doing that we'll have a paper out soon on, but I didn't include in this talk because I have too much stuff already, is you can do things which are very analogous to conventional medical MRI, so-called phase encoding or Fourier space, K-space imaging. I just threw several buzzwords out there. They're all the same idea. An MRI instrument, when Mike Crescimano goes in to have his bursitis looked at or whatever it is, and they put him in the MRI instrument, they get out an image that might have about 1 millimeter spatial resolution, but the wavelength of the radio waves that are coming out are close to approximately a meter. How are they able to do that? It's kind of near field detection in a way, but that doesn't explain it. It's not that they're scanning around with a little detector all over his body. They're detecting simultaneously for the entire body. The way they do that is the so-called phase encoding or Fourier imaging, which is they apply magnetic field gradients. So they tip the spins over. There's Mike's body, and they create with a magnetic field gradient, they create a modulation in the phase across the field of view. And then, if Mike's body was uniform and had no structure to it, and they just sat back and detected all the signal, all those modulating phases would cancel to zero and you get no signal. But what if Mike's body has structure to it? A lot of Mike is in this direction, and a little in this, and a little in this, and has some periodic structure to it. The spatial Fourier component, and that Fourier component was equal to the K vector of the modulation you put on. Then you have a lot of magnetization this way, relatively little of the stuff that's pointing that way is almost nothing there, and then a lot in this direction, very little in there. You'd have a non-zero signal you detect because the K vector of the spin modulation you applied corresponds to a non-trivial actual physical modulation in the object. We can apply the same sort of techniques to NVs to do phase encoding and image. We've done this now with 1 nanometer spatial resolution, many NVs across the field of view, and map where every single NV is, and then also image the patterns of magnetic fields that we're interested in using those NVs. Now, you do want the NVs tightly spaced so you don't have big dark regions, and that is another kind of limit to your sensitivity. Otherwise, you'd have high resolution but patchy imaging. There's things going on with compressed sensing and a lot of other techniques too where maybe Paolo's going to talk about that soon. So let me stop because I've already said too much.
Thank you.
Yep.